Now you have seen how purines are formed. Now let's come to the next topic, that is how pyrimidines are formed. One of the major difference between purine and pyrimidine is that you saw that in the case of purine synthesis, the purine ring is formed after we have already formed the ribose sugar moiety. But here the ring of the purine base or nitrogenous base is formed before it's connected with the ribose moiety. So this is one major difference. The sources of atoms again are almost the same as was the case in the case of purines. Glutamine gives rise, glutamine and aspartate, two major amino acids, they give the nitrogen moiety, nitrogen group to these compound, these nucleosides or these nitrogenous bases, and carbon dioxide gives the carbon part. Then also you should keep in mind one of the very important reaction here is that synthesis of carbamyl phosphate. This carbamyl phosphate is synthesized under the action of the enzyme carbamyl phosphate synthetase. Now this carbamyl phosphate synthetase is known as CPS2. So carbamyl phosphate synthetase 2. And you have, if you remember, it must struck your mind, your brains, that you have already heard something like this. Yes, you are right. That was the CPS1, carbamyl phosphate synthetase 1. And when we talk about it, when we are talking about the urea cycle, and you know that urea cycle takes place in the liver, in specific and major part of this reaction takes place in the mitochondria. But in the case of the action of the CPS2, that is the synthetase which is involved in the formation of carbamyl phosphate for the formation of pyrimidine, it takes place in the cytosol. Then after you have formed the carbamyl phosphate, then this carbamyl phosphate undergoes further reaction to give rise to the formation of carbamyl aspartate, and then eventually we end up with the formation of orotic acid. This is then changed into orthidine monophosphate under the action of orate phosphoribosyl transferase. From OMP, then you eventually end up in the formation of what you actually want, the pyrimidine base, that is the, or not only pyrimidine base, the pyrimidine nucleoside, that is UMP, uridine monophosphate. Once you get the UMP, that is uridine monophosphate, then it's then further phosphorylated to form the UDP and UTP, that is uridine diphosphate and uridine triphosphate. Here one important thing is that UTP, I really want you to focus on it, that UTP is also a very important precursor for the formation of DUDP, that is deoxyuridine diphosphate. You keep in mind that we have been talking about the oxygenated nucleosides which contain oxygen atom on the carbon atom position number 2 of their ribose moiety. But here UDP also acts as a precursor for the formation of deoxy-UDP, and this deoxy-UDP then can become a part of the DNA formation by making DUTP. But you should know that uracil is not part of the DNA, it's part of the what we say RNA. So one way the nature tends to minimize the erroneous or errorful incorporation of uracil in your DNA is that the DUTP is very readily hydrolyzed into DUMP. So there is no DUTP available further for its incorporation into the DNA. And this is what we have did here. So this is how nature have divides the ways to bring least sort of errors in the formation of your code, your DNA.